This is the International iAQA certificate on digestion. Digestion is a crucial role for supplying the glucose that, as we looked at in respiration, is an essential component for that chemical reaction to take place. So having looked at how we get the oxygen in for respiration, we're now looking at how we get the glucose in for digestion. Digestion has a key definition of breaking down large, insoluble molecules into small, soluble molecules. These are then able to be absorbed into the bloodstream, where you can then make use of them. For digestion to take place, there needs to be a series of chemical reactions that take place, and this is usually controlled by a whole range of digestive enzymes. You should know that in terms of organ systems, there's a hierarchy of size, starting with the smallest being cells, the smallest unit of any living organism, groups of cells of the same type working together for a common function, for tissues, groups of tissues all working together for a common goal, there's an organ, groups of organs would make organ systems, such as the digestive system, and then groups of organ systems make a whole organism. We're going to be focusing on a few tissues, but primarily the organs of the digestive system. In your notes, you have a copy of the digestive system. You need to be able to label those key parts. And the key bits that we have are seen here on this slide. There's a few more to label as well. Starting at the mouth, food is chewed, so physical digestion takes place. Amylase is released in the saliva from the salivary glands to help start the chemical digestion process of breaking down starch into glucose. Food is then swallowed down the esophagus, sometimes known as the gullet, where it will pass through a ring of muscle, and that ring of muscle is called a sphincter, where it enters the stomach. The stomach itself is another muscular bag that squeezes and churns the food around, squirting in all sorts of acids and other digestive elements, turning the food from the solid that we've started at into a much more liquid chine, as it's called. It sits in the stomach for a couple of hours, whereby it is then squirted out through another sphincter into the beginnings of the small intestine. The small intestine is divided into two sections. The duodenum here, the first part as you immediately leave the stomach, which is responsible for most of the chemical digestion, which we'll look at in more detail in a moment. And then as the food passes through, it enters the second half of the small intestine, called the ileum. This is where most of the absorption takes place. So we have chemical digestion, and here we have absorption in the ileum. From there the food travels into the large intestine, primarily known for its water and mineral reabsorption, and then the very end stage is the colon. This colon is for predominantly water reabsorption. Food, waste food, all the leftovers will be stored in the rectum at the very bottom before passing out through another sphincter called the anus. So the whole digestive tract runs from mouth through to anus. What you'll note is that the food has not passed through certain organs, but these organs are essential in the digestion process. They have not passed through the liver directly, nor the gallbladder, nor the pancreas. We'll look at the roles of each of these in the digestion process shortly. If you refer back to your notes, you will see that there's a table. In this table it tells you all the different acids and enzymes and other components of the digestive system which you need to know the functions of. This should already be complete, and it's essential that you are able to pick out any one of these elements and identify the name of the chemical or indeed where it is produced or any other feature for that matter of this table. So it might be worth pausing at this point to make sure your table in your notes is complete. Looking more specifically at the stomach itself, again this should all be in your notes. The stomach produces hydrochloric acid, which is a very, very strong pH 1.5 to 2. That is combined with an enzyme, protease enzyme called pepsin. And hopefully you'll recall from your enzymes work that protease enzymes break down proteins. Proteins are broken down into amino acids. Once they're small enough, they will then be able to be absorbed into the bloodstream. As already mentioned, the mixture of food with all these different gastric juices is ultimately called chyme, and it's that chyme that is produced as vomit when it comes back out of the stomach. From there, the food entered the first part of the small intestine, which was the duodenum. And the duodenum needs to work in a fairly neutral environment. So therefore, the strong acidic conditions that came out of the stomach aren't suitable for the enzymes. And so therefore, the juice contains sodium hydrogen carbonate, which is an alkaline solution to help neutralise that acid that has been produced. All of these chemicals are coming from the pancreas. So if we go back to our diagram, the pancreas, this leaf structure that was here, that's the one that the food has not passed through, but the sodium hydrogen carbonate, along with many other enzymes, are produced from the pancreas and squirted, if you follow the line, directly into the duodenum, neutralising any acids that have come out of the stomach, plus providing the enzymes for digestion. Bile must also be produced and provided. Bile enters the duodenum, but this time from the gallbladder. So again, going back to the diagram, here's the gallbladder, situated up with the liver, connecting onto the same duct that the pancreas was providing its materials into the duodenum through, and so therefore more neutralisation is able to take place from the bile produced from the gallbladder. The liver produces the bile, the gallbladder stores it and travels down through into the small intestine for neutralisation to take place. Bile has a crucial function. Refer back to your table. Bile from the liver stored in the gallbladder, not only does it neutralise the acid, but it also emulsifies lipids. Lipids, you'll recall, hopefully from your enzymes and molecules topics, are made of fatty acids and glycerol, and therefore they need to be broken down. But to aid the breaking down process, making it a lot quicker, we're breaking down our lipids from large globules into smaller droplets, so we have a much larger surface area, therefore meaning that the enzymes which will act to break down these lipids into their fatty acids and glycerol, have a much larger surface area to which to act, increasing the rate of digestion of lipids. So this emulsification happens from bile. So it's breaking from large drop molecules into small droplets. As a result of the lipase action, fatty acids and glycerol are produced, and therefore if you're in an experimental situation, this acidity would increase, so lowering the pH as a result of the fatty acid production. Once the food has passed the duodenum, it goes into the ileum. And the ileum is where most of the absorption takes place. So here is your digestive system again. If we focus in onto the small intestine, we can see that it, again, just as the lungs did in breathing, have the small intestine has an increased surface area. And that's achieved by not only being a highly folded structure, but each fold has many of these finger-like projections sticking out of them. And these are called villi. So one villus, many villi in the plural. Each one of these villi, if you look at this one, has its own rich blood supply, again, just like the lungs did, to have a rich blood supply over each alveolus. Rich blood supply to maintain a steep concentration gradient, maximising the rate of absorption into the body's blood system. The large surface area increases absorption. The distance that the molecules have to be absorbed across is reduced by only being a couple of cells thickness. One cell thickness on the cell wall of the villus, another cell for the capillary wall directly. So it's the same principles for exchange. Large surface area for each of these. Maximise exchange by a large surface area. Short diffusion distance. And also good blood supply. And the job of that is to maintain a steep concentration gradient. Crucial elements for the exchange of any molecule in living systems.